Okay, homology-based inference. This will be the topic today. So some general challenges for homology-based inference and what it is. Again, alcohol dehydrogenase is an enzyme that has part of a variety of aspects of its function. It's part of a pathway, it has a molecular function. Now, ultimately, let's get back to the word homology. What typically do you associate with the word homology? Yeah?
Knowledge-based inferment of function?
Yes, and this is sort of the first time you hear about the word homology, this is exactly what jumps to your head. Ron, fair enough. This is a very special understanding. So you took some lectures, and in these some lectures you had sort of a protein function structure prediction based perspective or skewed perspective on homology. But sort of more generically, if you had nothing to do with proteins, yes?
It's evolution.
Evolution, but what? What in evolution? Do you have any idea what the term is used for?
What the original definition of homology?
Yeah? Ron?
It's just functional similarity.
Of what and what? Yes, you're right.
Ancestral connotation.
Ancestral connotation, so you're already going a lot of steps ahead in some sense. Originally, really, it was, ancestral idea was there. Yes, there is the idea you have an ancestor. You have two from a common ancestor, and these two, you can see, have different organs or have some different body parts. The whale, as you all know, the whale at some point was living on land, moved into the water, so came out of the water. Some species went back into the water, and upon moving back into the water was a mammal, obviously, still is a mammal. And mammals typically have 4 legs, and the whale does not really have 4 legs. So somehow the legs have gone somewhere. Where have the legs gone?
They've turned into fins.
For instance, yes, so that is the term originally that was meant by homology. So you have fin/hind leg, right? This is sort of the homology, this is the relation. You have something that switches, and through that switch you recognize that there is some common ancestor who had the 4 legs. And that is what they have both in common. And you see how what the leg in one is the fin in the other, right? That's the idea, and it brings us back to something like a common ancestor. It brings us back to, in the simplest version of it, a tree. Today, we know trees are sort of misleading because we begin the more we sequence, the more we actually break down the concept of species. That's sort of the crux at the end of the day. The most important point really is species is, biologically, how is species defined? It's a very simple definition, yes?
It's individuals that are capable of reproducing each other.
That's not quite enough. Reproducing...
And that have offspring that can reproduce.
That's the crucial bit. That's the biological definition, exactly. So the idea is that the hybrid will in fact not have kids, right? So the hybrid is there, but the hybrid will not have new kids. That's the idea of the species barrier, so to speak. And we know that there are exceptions. There are tons of exceptions. We know that we carry Neanderthal gene. That is one example. So we are essentially hybrid. We are survivors from hybrids. We know that, I don't know whether you know, since we were on the issue of whales, narwhales and beluga, for instance, they can have hybrids who will have offspring. That's one common example. And narwhale looks very different from a beluga. So you may not immediately suspect it. So there are particular pairings of whales where it works, others not. It's not that any pair of whales can have these hybrids. But the more we look at the genetic reality, the more we look at bacteria, we know it from plants. But ultimately, with the genoming, we are realizing that essentially this idea of hybrid, the idea of species, is something that ultimately will disappear. We still hold on to it because it helps. It helps to make trees, it helps to think about things, but ultimately I believe it will disappear. Nevertheless, so that is one problem. But I'm going to talk about this sort of related problem. Now, the crucial issue here that I have on this tree, there is a big jump from what we are talking about and what I'm showing on this tree. On this tree I'm not showing species, right? I'm showing ADH1, so it's a particular gene, a particular protein in human and plant. And I'm building a tree for a protein. Now, whatever we talked about evolution is on the level of a species. The limitations on the level of a species. Now we're talking genes. There the story is getting yet more complicated. Nevertheless, these are essentially the trees that on the molecular level you build. Okay? Now, on the molecular level, you would then say that there is some common ancestor here, ADH1, and you assume that this common ancestor had some molecular function that is common to the one that we have today in human. Okay? Homologs have a common ancestor is one way in which we sort of talk about it today. Again, it's a misnomer. It's a change of the original meaning. But that's one way. And then we sort of adopt this to another level and say homologous proteins are proteins where the species have some common ancestor. And they had a common protein or gene. And that is sort of how we, in some sense, misuse the word homology. There is, of course, this part of the problem that we are not really clear that the tree is rooted. In fact, we are clear it's not rooted. And it's also clear that what we know today is absolutely not this rooted tree where there's a linear behavior. It's much more cloud-like, not only on the level of the organism, but much more so on the level of the proteins. For each gene, we cannot really see which species it came from. We see there's sort of a bunch of cloud effect kind of things. Anyway, now let's just, so the tree is a part of this. Now, orthologs, in fact, is the term that many people use for the subset of genes or proteins in this tree that essentially go down the line from the common ancestor. That are the ones that you would assume the common ancestor had this ADH1, we have this ADH1, and it did the same thing. Okay? Those are then called orthologs. They have the same function. Do you happen to know what the ones that do not have the same function are called? Those are the paralogs. They're divergent functions. Why could they diverge? The idea is, well, you have a second copy here. ADH1 already can do this original function in this plant here. You have another copy, ADH2, and since ADH2, in fact, does not have to do the same function, it is free to evolve something on top, something new. It can be a backup copy, so it can still do the ADH1 function, but possibly can evolve to do something else. And the moment it evolves to doing something on top, immediately you see that it has an evolutionary advantage. Right? And this is, in fact, the way we sort of see evolution happen all the time. Again, these words, ortholog and paralog, assume that we can write down this tree and clearly define function. So there's one particular feature of function that I can trace in the evolutionary tree, and I can distinguish between paralog and ortholog. In reality, it's not as simple. In reality, again, different aspects of the Go tree or of the Go hierarchy, whether you're looking at molecular function or pathway, they may actually differ. So one function may be the one from the ancestor, the pathway part, while the molecular function may have changed, or the other way around. Okay? So it is a little bit less clear. Now, then there may be another ADH2B that in fact is not coming from a common ancestor, but it still may have a similar function. How can you imagine that? So you could have a protein, I could even call it ADH1B here, that would have been maybe the better term to use, that has some similarity to this one here. It's called plants, yes. There's some similarity to this. It's not coming from a common ancestor. You may infer that it has a similar function. How could that happen? Yeah?
Is it possible that depending on where you are, like I'm wondering if it's like with insects and birds have both developed wings, and I'm wondering if this also works out on the molecular level, that you want to fly.
Certainly
so
does.
You want to have something similar. You want to do something similar, so we will have.
So certainly something similar. But before we get into thinking about a bird, let's make it much simpler. You want to create a certain bond In order to create a certain bond, you need a certain structure to stabilize that bond. In that sense, you may call it just like in order to fly, you have to invent the same principle. Here it's the same bond, and you could get to the same bond from slightly different parts. You could call that convergent evolution. So you could come from sequences that originally were different, and they just evolved to the same function simply because there's one way of doing the bond. So it had to adopt, in order to do that bond, it had to adopt this particular three-dimensional shape or this particular local three-dimensional structure in order to do that. Yes, exactly. And there's a lot of that visible. It's very difficult to distinguish when ADH1 and in this particular case ADH2B are so divergent in sequence. It's very difficult to say, is it now from a common ancestor? Or is it something that was adopted anew? Is it a paralog? Or is it convergent evolution? It's difficult to argue that in detail. Anyway, so just to get back to these words, the tree is uncertain, the story is complicated, we are on the level of proteins. On the level of proteins, they don't reproduce, and there are a bunch of additional problems. And the first and biggest one essentially is the domain problem. The domain problem is, in this particular case here, we see that protein A and B have a domain in common, B and C have a domain in common, and if we had the annotation for A, we could not say anything about the function of C because they are not sharing the domains. Now, on the level of where you have on the level of where you have functional annotations, you typically do not have domain annotations. So typically you say that a particular protein, ADH1, is cutting alcohol or is processing alcohol in some way. And typically in most of these annotations you do not know, well, the part that does the alcohol is exactly that domain. Because for most proteins, again, the part, we don't quite see the domain. So that makes it difficult to sort of do homology-based inference without acknowledging domains. And since in many cases we do not have domains, homology-based inference becomes a little bit tricky. The other problem is the moonlighting problem. Who in the group has heard about the word moonlighting? Moonlighting, yes. So ultimately, it comes from people who have essentially two jobs and have a second job, a second full-time job in the night. And again, in New York City, this is quite common. We're talking about proteins. What do you believe it could be? So they're moonlighting proteins. What could that mean? Yeah?
In one case they have one job, and in another case they have another job.
Exactly. And this, of course, makes it extremely difficult to infer function. Because now what could be the reason that they have one job in one case? So this particular case is not about shortage of money for paying rent. For proteins, there is a sort of a related reason in some sense. You have to pack it into a small genome. But what could, yeah?
Yeah, I think where it's expressed.
That is one important thing. So you simply see that by being expressed in different types of cells, you have a different function. Why could the different environment make you function differently? Yeah?
Because some binding sites not only bispecific, but are rather open to many kind of
Why?
bindings, because they're not rigid enough.
No, but so if they are open to many things, so why would it differ between whether you're on, say, a muscle cell or a neuronal cell?
Because you might have to digest different enzymes, different substrates, for example. So the function is quite the same, but rather for different.
Yeah, but why do you have, I mean, so who tells so you tell the enzyme, okay, you're now a muscle cell, you have to do this, or what?
That's genetics.
What do you mean genetics?
Epigenetics, is how it's regulated.
Oh, let's go before we have epigenetics, much simpler. You can explain it already much simpler, yeah? Yannick is certainly the, yeah?
Maybe it's the same, but I want to say maybe exon skipping, like certain parts or certain cells.
Yes, so that ultimately would get to regulation, but simpler, much simpler, yes?
Just like pH, depending on the
simplest.
environment.
This is simplest. So even in the same cell type, it's different subcellular localization. You could, when you begin saying that the binding site is open, This could actually be influenced by a different pH value. That could be one reason. What else could be the reason? Also simple.
Ligands?
Ligands, exactly. But why would the ligands not be in one?
Well, it really depends on what else happens in the cell when something else is produced.
Exactly.
Yeah.
Exactly. So this is long before the regulation. So it could just be that you are sort of binding DNA because you're in the nucleus. In the nucleus you encounter DNA, while when you are in the plasmic reticulum, there's no DNA floating around. You're not going to bind DNA. As simple as that, right? That it could be that in the different cell types you have different protein binding partners. How could that happen? Why would you have in a muscle cell different? Well, you're going back back to regulation. So essentially, this is part of regulation, but it's still not epigenetics. So different cells express different parts of the genome. As simple as that, right? That means the possible interaction partners may just not be, or may just be different. You bind to two different proteins, and one you find in muscles, and one you find in neuron cells, or something like that, right? You immediately could imagine a simple mechanism, far away from regulated, a simple mechanism by which you explain moonlighting. And in fact, there are many examples of moonlighting proteins. The first examples of moonlighting proteins are of the simple type. All the complicated types that you're talking about, alternative splicing, epigenetics, they do happen too. But it's something coming on top, okay? And I would still not call them moonlighting, because that actually, so the exon, the change of the exon would change the protein. The epistatic is a slightly different, yes?
So are moonlighting proteins one protein
Yes.
that does that, exactly that protein, like that physical molecule,
Yeah.
does two different functions?
Yes.
Or is it a protein that is expressed and in one environment it does one function, and in a different environment when it's expressed it does a different function, but it's never exactly the same molecule? You know what I mean? Like if we were to follow this one protein, would we see it do two different functions? Or is it, oh, we're looking at this protein and it's expressed in the nucleus and it does a function, and oh, we're looking at it in...
Let's call it a little bit of a hypothetical question, because it's difficult to push one protein into different cells. Unless you go through the biological pathway, in the biological pathway the same protein is not expressed. I mean, you cannot have expressed life begins, and you cannot express it again, okay? So you cannot express it in a muscle, but assume you would push it through. So suppose we would get in vitro. Let's suppose we would be in vitro. And I would put that protein into two different cell environments. They would indeed. The same protein. Again, we cannot do this, the experiment with one protein, but assuming that I have this protein expressed, and now I take whichever one of those I have expressed in there and there, I essentially have the same situation that you're talking about. I have the same protein.
Yeah, but that's a contrived, so I guess the question was if that happens.
But the assumption is then that it's the same, right? The assumption is that the answer is the
Well,
same.
it's the same molecule, it has to work in both, right?
That is ultimately the assumption, yes. Yeah, yeah. Okay, now the concept of homology-based inference is, you're way ahead of me, is very trivial. You have one particular protein here, this is an alignment here, one particular protein for which you have an annotation, one particular protein for which ingredient for which we know that it's similar to the one for which you have the annotation sequence similar above some threshold, and if the sequence similarity is above some threshold, you simply say that this protein has the same function as the first one, right? And in this example that I pull out here, let's say I want to infer that a particular protein is a methyltransferase. I'm going to show you a couple of examples here, and for these examples the blue ones mean that I get it right. So say I put my threshold for what I call similar enough in sequence at 100% sequence identity. I'm sort of right, forgetting about moonlighting protein. I'm putting my threshold at 99%, I'm getting right. And then sort of putting it at 70%, I see I get the first wrong one here. It is a transferase, but it's a formal transferase. It's not a methyltransferase. I get further down, so this means that there's something below the 70 that still is a methyltransferase. So it is just there are outliers here, and if I stopped here, then what I essentially would get is 2 out of 3 right, and 2 out of 4 that I could have. So sometimes this is referred to as precision, and this is referred to as recall. Now, if I sort of went further down the line here, I may see something, it's a completely made up example, I may see something like this. And that triggers the question, what is the better threshold? Is it better to stop here and have 2 and 3 right? But finding 50% of all those you could have found? Or is it better to stop here and have 3 and 8 right, but finding all that you could have found? So here's the same question back in number. So what is better? You're smiling because I asked you the question before?
No, but it's depending on the question, I guess. I mean, if you want to find all methyl traces, you need to lower the accuracy, but there's no way of telling.
There's no way of telling, exactly. So ultimately, it depends on it depends on what you're after. It's no clear. So now, say you developed a method to define the threshold that helps you to infer the first one I'm going to show here. My first specific challenge will be to predict subcellular localizations through homology. So which one would be better?
Depending on the downstream analysis I have, when it's quite cheap or something, I would go for the lower accuracy to find out.
So let's revive that question when we see the data. Is ultimately what you say in your first sentence, right? Depends a little bit on how it looks. Okay, so this is work that goes back to Regis Nair, and ultimately we're trying to do subcellular localization. This is a eukaryotic cell, the different compartments, and essentially in the simplest way, I believe the original data from Regis really looked at nuclear, cytoplasmic, extracellular, so in a very simple way. And we're trying to, just like we did last semester, we're trying to see when I come from this high sequence similarity to low sequence similarity, so from the zone where I essentially assume that 2 proteins clearly would have the same subcellular localization if they have enough sequence identity. At some point I will sort of lose the signal, and at some point I will not have a signal at all. And what I do care about is essentially how is the transition from one zone into the other. So if I began with high precision, then precision will drop. Will it drop that extremely? And that's the recall. That will be the opposite to that. But exactly how it looks, I want to know. What do I need in order to establish these curves? What do I have to give you so that you can actually make that happen? That you can plot this? I have to give you data. What?
Multiple sequence alignment.
What do you need multiple sequence alignments for?
So that I can threshold the sequence similarity and calculate.
Okay, so I give you a bunch of multiple sequence alignments. Is that enough?
You would need to know which one are indeed all available transferases in this.
So we need a bunch of proteins for which I know the subcellular localization. So we need annotations. Okay, so I gave you proteins from a database where you can look up subcellular localization. It's for instance SwissProt. There are other databases out there, or that is one of the better ones. So I give you the entire SwissProt. I give you the subcellular localization for all the proteins in there, and then you sort of produce for every pair. You do need some, not necessarily alignments, but pairwise sequence identities, right? The minimal level, you just do an all against all of whatever has an annotation, which is not quite a multiple sequence alignment yet, right? You could argue you could do it better, and we will get to that, but let's look at the first step first. Is that the way we would do it? Okay, let's turn that around. Why is that not the way? What's wrong? I give you everything, you take SwissProt, you pick out everything you find in SwissProt. Why is that wrong?
Probably because it's pretty huge and too much data.
Actually, it's not that huge. So, in fact, I do not know if what the current number is, but it's in the order of ballpark of 20,000, 20,000. A little bit more, yes?
It's your bias.
Sorry, this was Thomas behind you.
Just not raising my hand very often.
Yes, you do, but the point is that when I show there's no difference for him to see whether I might mean you or him.
So I think we are not considering like evolutionary couplings and...
No, we are not considering them. In fact, Paul wants alignments. I'm not giving him alignments. I'm clearly not giving you evolutionary couplings.
Okay.
That is many, many levels beyond. No, but there's something else wrong here.
biased because in the
Yes.
infinite zone we will not find those proteins.
It is biased, and I'm going to show you how that will hit you, that is biased. Maybe not today, but maybe on Thursday. But what you actually have to do is you have to have a subset of proteins that are sequence unique, that are sequence non-redundant, that are such that there's no pair in that subset that are highly similar to each other, in order to get a sort of sample of unrelated proteins to start from. So now I'm going to do this one against that one. Why am I not doing this one against itself? So that's the least redundant. So again, let's explain again how we do that. We take the 20,000 from Swissprot, we pick the first, say you're on the list now. And then we go through the list and ask, we have the alignment of all against all, 20,000 against 20,000. And we take protein 2 in the list, and we ask, is protein 2 highly sequence similar? Say 90%. If so, I kick it out. I take only things survive that are such that there's no clear sequence similarity. That produces a data set that is best at representing new proteins. Because it's something, I will try to do this for new proteins. And this is a little bit more complicated. I believe this is the best way to sample what I believe will be a new data set. Okay? Because put it differently, there is a lot of redundancy in here that comes from the fact that people, if one person has shown that a particular protein is nuclear, to show that the same protein is nuclear in a very similar species is the simplest experiment you can have. And you get an equally good publication almost, right? So that's one incentive to bring a lot of proteins from very similar with very similar sequences and redo experiments. Another reason is, if you had, for instance, a protein in mouse that you know is nuclear, and now you have a disease in human, and you know the protein, you believe that the protein in human is actually related to a disease, and you think about developing a drug, you're not going to believe simply because mouse protein is nuclear and the human protein is nuclear, they will both be nuclear. Before you invest into drug design, you're actually going to check whether the human protein is nuclear, right? This is another reason why you have a lot of redundancy in the real data set. This redundancy you have to kick out because you want to sort of have an non-redundant way of getting your curves. Okay, you have to begin with a non-redundant subset. You always have to begin with in biolecture you have to always begin with a non-redundant subset. Let's keep it here. I will show you some arguments later. But why can you not do, if I have this credo for non-redundancy, why not do non-redundant against non-redundant? It's totally trivial. If you believe that you cannot answer the question because it's too trivial, just answer it. It is totally trivial, if you know it. Yes?
Because then you would essentially neglect biology behind it.
No, that's not the point. It's much simpler. That's not trivial. Yes?
Pretty much can't expect matches then, can you?
That's the issue. That's the real issue. If you want to have a curve that shows how you get from high similarity to low similarity and then see something like precision and recall, if you have 0 hits, you're not going to have a curve. Right? So this is the problem. You have to accept the redundancy on one end, otherwise you have 0 counts, okay? Which is, you have to somehow accept redundancy. You could possibly filter that by again saying at least 100% you kick out, or 99% you kick out, or something. For PDB, just getting back to that point, for those of you who have been here in the last semester lecture, the PDB, the database of protein structures, at this point is about 200K. If I just filter out 100%, so meaning that I go through protein by protein and I kick out whenever I only let things survive to the ones that I already have in my stack that are less than 100% identical to what I have, right? 99% I accept, 100% I throw away. And already at that point I get down to 80K. So this minimal reduction one possibly should do here, given that some databases are really that redundant in biology. For this particular case, it's not that much of a problem. Anyway, then you get this. Now, unlike the way I usually draw that, no, I changed the colors in this particular case. Now this is that's structure. For structure, number of residues aligned, percentage sequence identity, and again, some of you have taken last semester's course. This was a slide that I showed very often. There is the H-val, the HSP curve here in gray. The HSP curve is such that essentially there's no red point above the gray line. There are some. This is biology, there are always exceptions to that. But essentially above that gray curve, you have only blue points, right? Now, I do the same for subcellular localization. Can some people I'd like everybody in the room to say something about this curve. And I'm going to put off the camera. Daniel, right? Okay. So here is the transition for this is recall, precision. If I plot the percentage sequence identity, here's the same thing if I do e-value, a psi-blast e-value. And here's the same thing if I do distance from the gray curve that I showed you. Is it clear what I'm saying? Is anybody in the room who has an issue?
Can you repeat that?
So what I'm going to show you now are three different curves. One is simply going to ask, so I'm going to change the way I represent that. I'm just going to find a way to measure sequence similarity between two proteins and compute how many are blue and red at a given level of sequence identity. And I have three values for sequence identity. Percentage sequence identity is the simplest. A psi-blast e-value is the expectation value that comes out of a psi-blast or blast alignment. And the third one, essentially this e-value from psi-blast, so you have a background distribution and you have a score. And you ask, how far is the score from the average of the background distribution? The background distribution means essentially random alignment, so to speak. How far is the score, the alignment of the pair, different from the average? And what is the standard deviation? Right? Those two values essentially go into computing the e-value, and I'm going to show you the e-value, and the third one I'm going to show you how different is the pair from this gray curve, where the difference essentially goes in two directions. It could be above 0 and below 0. So plus 5 would essentially be a parallel curve plus 5. Yes?
Well, why don't we shift the curve a little bit?
As in with a different expression, so to speak?
Yeah.
Because this was simpler to just do plus 5. Is that what you're asking?
No, I mean, because like on the left it looks like the curve fits it quite well.
Yes, but the problem is here, so you see,
have a lot more outliers.
so you might argue that you just move it up here, but I didn't want to have another fit. As simple as that. I felt that one fit is enough in my life. In effect, again, so this is a highly quoted paper. So many people have come up with the new fits. I'm not sure whether they have done a fit for this one, but there's a more recent fit for functional groups. But again, it sort of moved the original. I believe you could also come up. Anyway, we did not do that at this point. Why not? Because we were lazy. Let's give that as an answer. Yeah. No, we cannot give that as an answer because it was simpler. Because Rajesh didn't have enough time. Rajesh clearly is not lazy. Anyway, everybody understands the three curves that we're going to have? Those are the three curves. We have two lines. Essentially, one measures recall. So how many of the ones at that threshold that I could have found did I find? So I have a set of proteins that I know the localization for, so I know how many are nuclear, I know how many I could have inferred. That is the line that goes up. The recall goes up, while the precision goes down. Precision, how many of the ones that I find at that threshold are right, are correctly predicted. So above, say, the HSP distance, and here the HSP distance is, you see, negative. Here's 0. 0 essentially is the line is here. And you see that the line is not quite right, but it's not such a bad point in terms of meeting where things go into a rapid decline. So, which one of these three scores is best?